So now I'm going to introduce Dalia Goname. So Dalia is a PhD candidate in biomedical genetics at the University of Rochester Medical Center. And today she will be talking about machine learning and identifying non-coding RNAs.
Thank you, guys. I just wanted to say thank you for the opportunity to talk about this. This is actually my first time presenting on this specific topic. So if you guys have questions, please stop me in the middle, because I don't really have much experience in this, talking about this topic to know what is confusing and what's not. So, so I'm excited today to talk about machine learning and identifying non-coding RNAs. So my goals for this talk are that by the end of this lecture, you'll have an idea of what machine learning is and why it is exciting. And also, I hope that you have some exposure to what non-coding RNAs are and how machine learning can be used to identify them. And the majority of my talk will be about machine learning, and then this is more of an application for how it can be used. So can I see a show of hands of how many of you have heard of machine learning, even if you don't know what it is? So a few of you. So machine learning is the scientific study of algorithms and statistical models that computers can use to perform specific tasks without explicit instructions. So basically, it's using computers for pattern recognition and learning patterns without explicitly giving computers a function. So I'll explain more what that means if that doesn't make sense yet. So I have a simple task here. Given these three different input and outputs, I want you to take a second and see if you guys can predict what the input of 6 would, what output input of 6 would give. You don't have to say anything. You can just think about it in your head. So this is what the output would be. Did any of you guys get that right? Yeah, I assume most of you guys did. So I didn't tell you guys any function. I didn't tell you guys when you input 1, the function that you would get as an output. But you guys figured out by yourselves what an output would be if I gave you the number 6. And you did this by learning patterns from this data, determining a function yourself to predict what the output would be for 6. So you guys did this kind of by yourselves, but how would you get a computer to do that, do the same thing? So I'm going to talk about, that's what I'm going to be talking about. So there are 2 types of machine learning, basic types. The first one is called supervised learning, which infers a function based on input and output pairs. That's what you guys just did. There's also unsupervised machine learning, which is when you have a data set that doesn't have labels. It's a little bit more complicated and involves finding patterns by clustering data, creating networks, and kind of trying to reduce the dimensionality of data. I will not be talking about that, but I wanted you guys to know that there's other kinds of machine learning. So going back to this example, you guys took a training data set and inferred a function, and then were able to predict a new output for a new point. In machine learning, it's important. Machine learning works really well when training data is good. So if I had given you a data set that only had 1 point, it would have been really harder for you guys to accurately predict what this would be. It also assumes that new points follow the same rules as old points. So for example, you guys had no idea what the 6 could be. It's possible that the 6 follows a totally different rule. And machine learning does not work in these cases. It's important that the training data is good and that the new point that you add to it follows the same rules. So I'm going to be trying to cover quickly 3 algorithms for supervised machine learning. I wanted to mention a couple other ones, k-nearest neighbor and decision tree. I'm not going to be talking about them, but I want you guys to hear the names in case you ever decide to look further into this so you recognize these names. The first one I'm going to talk about is called linear regression. It's probably the simplest method, or the easiest to understand at least, and many of you may have done it before without realizing it. And I'm going to apply it to a really kind of simple, maybe silly scenario. Imagine you're at a little cousin's birthday party. You don't remember how old he is. You're too embarrassed to ask. So you have access to a training set of ages of boys and their heights, and you want to predict the age of your cousin based on his height. It's probably easier to just ask, but so assuming that you have this training data set, and I'm sorry it's blurry, of heights and ages of boys, can you determine a function that can predict the age of a boy based on his height? Well, I plotted this training set, and maybe the easiest way to do this is to find a line that explains the data. For example, here's a line that explains it, but I could also draw another line, and how do you know which line is better than which? So the way linear regression works is it creates a linear function that tries to minimize coefficients of residual values. I'll explain what that means. So let's say that you have 3 data points in blue, and you want to draw a line that explains the data points. And the line has these coefficients. In this case, it's the slope and the intercept that you guys have probably learned in math. And the way that it determines the best fit line is by trying to reduce the distance between the real data points and the data points that you would expect from the model. So if you take all these distances and try to reduce them, you can get a best fit line. And that's how linear regression works. So in this case, we used linear regression to predict the best fit line for this training data that we had. And the coefficients that we have are negative 11.9 and 1.57, which correspond in this line to a slope and an intercept. So if you look at your cousin and he's 108 centimeters, you can predict from the line that he's about 5 years old. And even though this seems really simple, it's actually pretty powerful. It is a method of machine learning. And in science, regression is often used to study how multiple mutations in genes are associated with diseases. It can also be used in chemistry to determine chemical concentration of analytes in solutions where the signal is distorted. And it doesn't have to be linear. So you can also do regression in a nonlinear manner. And it's a little more complicated, but it's also very useful. So the next kind of machine learning that I wanted to talk about is naive Bayesian. And here's another simple problem that you might have. So let's say you receive a text from a number you don't recognize. For simplicity, you only have 2 friends, Jeffrey and Kiki. And how can you determine who sent it? So here's the message. Hi, it's me. This is my new number. Weather's absolutely amazing. Meet at the pool. So let's say you have a training data set of old texts from Jeffrey and Kiki. Each text is labeled, either belonging to Jeffrey or either belonging to Kiki. Can you use this training set to classify the new text as either one from Jeffrey or one from Kiki? I'm trying to keep this simple. So this is the text up here. So let's say you scan all the messages. And in this example, we're only using one word, just to keep it simple. Let's say you want to use the word amazing to decide if it's Jeffrey or Kiki that sent this. Looking through all your text, you find the probability that Jeffrey would say amazing in a text is 0.2. The probability that Kiki would say is 0.3. You also find that the likelihood that Kiki would text you in the first place is 0.4, and the likelihood that Jeffrey would is 0.5. And the likelihood that any of the texts you would receive contains the word amazing is 0.2. This is important because this is a Bayesian formula. So I don't know if you guys don't recognize this, the way this is written, I'll explain what it is. So what this means is the probability of A given that B is true. So if you want to determine the probability that A is true given that B is true, it can be determined by the probability that A is true given B, or sorry, that B is true given A is true times the probability of A over the probability of B. This is a well-known statistical formula. We won't try to prove it, but so in this case, given all those numbers that I kind of gave you that were determined from a data set that I kind of made up, so if we want to know the probability that Kiki sent that text, given that the text contains amazing, we would take the probability that the text contains amazing, given that Kiki sent it, times the probability that Kiki texted you in the first place, divided by the probability that, that any text would contain the word amazing. And given those numbers that I had in the previous chart, the probability that Kiki sent it is 0.6. We can do the same thing with Jeffrey. We find the probability is 0.5. So given this model, you can predict that it's more likely that Kiki sent the text than Jeffrey. Does that make sense? Kind of. And this has a lot of common applications. This is what is used for spam filtering. So if a spam filtering software will check words in an email and decide, well, if this email says the word Viagra, what's the likelihood that it's spam? It's also used for predictive text. So I don't know if you notice sometimes if you text, maybe your keyboard will try to detect what the next word you're going to say probably is. Similar machine learning. Also can be used to determine if you have certain genetic mutations, what your risk for genetic disease would be. Okay. So the last machine learning algorithm that I wanted to talk about before we go into an application for it is called a support vector machine. It's a method for binary classification. And what that means is taking a group of data and trying to classify it into two different classes. And how it works is you have a labeled training set, and I kind of made this up. Like, let's say we have two different cell types, and we measured the expression of gene A and gene B in each of the cell types, and we want to be able to look at a random cell and try to predict which cell type it belongs to. So you can see here that the data separates, and just by eye, like you could draw a line that kind of separates the two data points. And that's what support vector machine does. It tries to create a hyperplane, and I'll explain more what that is, that divides two different classes. So the kind of, I don't know why this has all these things, but kind of complicated part, the kind of complicated part of a support vector machine is determining where that plane should be. Like you can see that this line separates the data, but so does this line. So which one is a better classifier? And the idea is, like, if I have these two cell types, if I take a new cell and see where it lies, I can try to figure out which one it belongs to in this example. So the way a support vector machine determines the best hyperplane, or the best line, you can think of it as a line or a hyperplane, is by maximizing the margin. So this line has a larger margin than this one. So the support vector machine would choose this hyperplane as a way to classify the data. And the support vectors in the name are these points that are closest to the margin, because they're the ones that determine the hyperplane. These are the ones that are important. The other ones really aren't playing a factor. So this was a simple example. It's just a line. But what if you have data that's more complicated? And this is kind of where the support vector machine becomes more powerful. So imagine you have data like this, where you can't really draw a line that separates the data. So what you can do is draw a line that is not straight and project it into a higher dimension to separate the data. So here is two different data types. If you project it into a third dimension and create a hyperplane, you can separate the two data points. And this is done mathematically through transformation of the data. It's called a kernel transformation. Here's another example that's kind of cool and complicated. So here's data that's also, you can't use a line to separate it, but if you project it into three-dimensional space, you can create a plane that'll separate the data. So those are the three types of machine learning that I wanted to talk about, and now I wanted to talk about a way that you can apply a support vector machine to identify non-coding RNAs. Okay. So I know that you guys come from different backgrounds, but genetic information is stored in DNA, and when it's time to express a protein, genetic information is copied from RNA and translated into protein. And the central dogma of biology suggests that RNA is kind of a passive intermediate between DNA and protein. But it's been shown that RNA plays more active roles in catalyzing reactions and regulating genes. And RNA sequences can be involved in cleavage, transcriptional gene regulation, and defense. And RNA sequences that are not translated, so not these ones, the ones that are over here, play many important roles in cells, and many of them have not been characterized. So it's, many people are interested in identifying them. So once again, these non-coding RNAs are important in biology and can cause disease if they're regulated incorrectly, and most of them have not been identified. Not all of them have a function, and they're difficult to identify because they evolve rapidly. And what I want you to take out of this is that they're difficult to identify and that people are very interested in identifying them because they play important roles. And I want to show you an example of how machine learning can be used. So how can you identify functional non-coding RNAs versus ones that aren't functional? Well, RNAs that have a function often fold into shapes that determine their function. So if you think of any object like a fork or a spoon, it has a specific shape that determines how it functions. And in the same way, RNAs fold into specific shapes in cells that make them have functions. And the thermodynamic stability can be used as a way to determine if RNA will fold into different shapes. So despite the fact that non-coding RNAs don't have sequence conservation and they're not, and they evolve rapidly, it's been found that functional non-coding RNAs tend to be highly conserved between different species. So this is just an example of a program that uses a support vector machine to classify sequences as functional non-coding RNAs or ones that are not functional non-coding RNAs. So the way it works is it uses a support vector machine on a data set of known non-coding RNAs, a data set of over 22,000 non-coding RNAs, and it labels them as either functional non-coding RNAs or ones that aren't functional. And then they trained the support vector machine based on 3 different features. And the 3 different features are like 3 different dimensions in space. So one of the features is the conservation of the structure, so how well it forms a structure across multiple species, and then how well it folds into a stable thermodynamic structure, and how poorly it folds into other structures. And they applied this to unknown data to predict new non-coding RNAs. And I kind of wanted to show like the way the space would be. So if you have non-coding RNAs, you're kind of separating them in space by 3 different features, like I said, the structural conservation, how thermodynamically well it folds, and how poorly it folds into other structures. And using these 3 different features, the support vector machine was able to classify non-coding RNAs that are functional versus ones that aren't. And it performs comparably well to other available software tools. And this is just one example of how a support vector machine can be used in science and specifically in identifying functional non-coding RNAs. But there are many different examples of how machine learning is used in genetics and chemistry and RNA biology. So in summary, machine learning algorithms use computational methods to learn information from data without relying on any predetermined function. And they range from very simple to more complicated, and they have many applications in science. Non-coding RNAs also play important roles in biology and have yet to be characterized. There's also many resources that can help you learn about machine learning, and they range from like beginner level to very advanced. And there are programs like Python and R that have libraries and algorithms that are already available for you to use to apply to your data. So if you guys find it interesting, I encourage you to kind of explore further. And that's all I have. So thank you for listening. Does anyone have any questions? All right, thank you. Oh, okay.
So how have you applied this with your research? Is it tied into your research?
Yeah, so I actually trying right now to identify non-coding RNAs involved in male infertility. So we have non-coding RNAs that we have sequence data from sperm, and we're trying to know which ones are possibly functional non-coding RNAs versus the ones that are just RNAs that are sequenced. So yeah, and people in my lab have applied this to many things as well. Yeah. Thank you.
Okay, thank you so much for coming. So this was week 1 of 4, so we're going to have 3 more of these. The next one will be next week, and we'll have 2 new speakers coming to present 2 new topics. So we encourage you to come back. We'll have food at every lunch, so please come back, and thanks for listening.